Welcome everybody for our second session for discussing the following paper, Phylogenetic Structure of the Prokaryotic Domain, the Primary Kingdoms, by Carl Woese and George Fox. So that paper is from 1977.
Yeah, I'd like to add that in our first of the series, we didn't mention the date, so it's Sunday, 13 February 2022. So for the record.
Yeah.
So last time we discussed the 1974 paper about that first talked about extremophiles, and this is the only 3 years later, and this is the classic Carl Woese paper about the three domains of life. Because before, up to this point in the biosciences, the fundamental distinction was between prokaryotes and eukaryotes. Eukaryotes being the more complex cell, which would probably resulted from endosymbiosis when one cell swallowed another, and prokaryotes being much simpler. And Woese in the paper argues for the prokaryotes are, there's more than one kind, so there's more than a fundamental division between two kinds of life, there's a fundamental division between three kinds of life. So the paper introduces a lot of new terminology, some of which stuck and some of which didn't stick. And it's interesting that since the Woesian revolution, as it were, prokaryote has fallen more and more out of use. So he introduces the term archaebacteria for what we now call archaea. So archaebacteria, that didn't stick around, but people kept the idea of a third kingdom, which we now call archaea. And what used to be called prokaryotes are now divided between bacteria and archaea. And then we still have the eukaryotes off to one side.
Yeah, what I really thought remarkable here that before him, they thought there is animals and plants, right? So, and they did this distinction between only two, but then they thought about, no, that cannot really be. And then they were looking for, because prokaryotes and eukaryotes are so distinctly different, so you cannot really compare them to make distinctions. And they were looking for where they can figure out what is the really difference. So the smallest common thing, and they came up with one part of the ribosome here, what makes the proteins. So that is the most common thing of both, and then they could see the differences because of these differences in the ribosome. So that's what they started here.
So the method that Woese pioneered is called molecular phylogeny. And in the paper, we see it says, quote, "An organism's genome seems to be the ultimate record of its evolutionary history," unquote. And I found a nice summary of the method of molecular phylogeny and Uprooting the Tree of Life by W. Ford Doolittle, who wrote, quote, "All genes mutate, sometimes altering the encoded protein. Genetic mutations that have no effect on protein function or that improve it will inevitably accumulate over time. Thus, as two species diverge from an ancestor, the structures of the genes they share will also diverge, and as time passes, the genetic divergence will increase," unquote. So this is about going into the molecules of the organisms and finding out the record of mutations and tracing it backwards to find where they split off, and discovering the life doesn't just split off into two kingdoms, it seems to split off into three. Okay, there we got our prokaryote and eukaryote cells with some elements in common, the cytoplasm, the mRNA, the DNA, but the one has a nucleus and the other doesn't. And here is a famous diagram. One of the terms that's introduced in this paper is the idea of the progenote, which is something even earlier, which could have been the common ancestor of the archaea, the bacteria, and whatever preceded the eukaryotes when they were simpler before they became a complex cell. And it's still not known exactly, you know, there has been no progenote definitely identified. There's a lot of theories, there's a lot of research that's been done, but there's no definitive answer to how, if any of the three kingdoms came first, or what branched off of what, or if there was a simultaneous emergence from different origins events. So this is how the
I'm still with the two cells.
Oh yeah, I've advanced a couple of frames. Maybe I have to restart it for each. Yeah,
but why it doesn't share? That doesn't make sense, because it shares your screen. That's really, I don't get.
Yeah, it's not advancing for you. So if I turn it off and restart it, then it might advance for you. So screen share.
Yeah. Okay. Now I have the kingdom of the three.
So this is a more recent depiction based on subsequent work to Woese, which shows the archaea and the bacteria and the eukarya as distinct kingdoms, with possibly the root in the center of the three diverging lines. Like I said, nobody really knows what came first. So I'm going to click this to the next slide, which you probably won't see since it's not advancing for you, but
there was actually a side note here in the paper that was actually very interesting, that they said there is a possibility that because of the symbiont theory, endosymbiont theory, that mitochondria and plus these chloroplasts, right, that they maybe come from other kingdoms which died out right now, and these are the last one representatives of that kingdom. I think I understood that right.
Yes. Yeah, I picked that up somewhere, but I didn't remember it.
Yeah, so that's very interesting. And he discussed also, do we find other kingdoms still? Can we find other kingdoms? And I think he was not quite, I mean, that's 50 years ago, right? But he said, yeah, it could be, but probably not. But yeah, when we find a different bacteria, for example, in the Lake Woodstock, right, what is millions of years isolated from everyone, so that could be that there are some. I mean, I'm speculating here, but that there are some other. Could be a domain, right? So
yes, he does suggest that there could be another domain, but in order to get the level to push it far enough back in time to be as fundamental as this, he notes in the paper what they knew at the time, that we're talking about lineages that are 3 billion years old. We don't know of any environments on Earth that have been isolated for 3 billion years. Now there may be some in the cratons at the center of continents that don't move much during the supercontinent cycles. That would be an interesting question to look into. But yeah, we do know of several ecotopes, I guess you could say, that have been isolated for millions of years or tens of millions of years. We don't know.
Yeah, that's not enough time. Yeah.
Okay, we don't know of any that go to hundreds of millions of years or billions of years. But like I said, they may be out there. Or if there's been panspermia in the solar system, there could be isolated bits on other planets that trace back between 3 and 4 billion years.
And there could have been more, but they died out over time.
Right,
So,
right. Because the biosphere has changed so much over 4 billion years that
yeah, and when you think about that, what is really interesting, because archaea and bacteria under the microscope, they look basically identical, right? So compared to eukarya or eukaryotes, what we have here, they are so different. We have plants and fungi and slime molds and animals, right? So they are vastly different. And bacteria and archaea are so similar in shape, but on the molecular basis, they are so different from each other, like bacteria from eukarya, like archaea from bacteria. So that's so interesting if you think about that. Yeah, I mean,
yeah,
when you think now, if hopefully in our lifetime we find microbial life somewhere in our solar system, that could be totally different, a different domain, even when it looks the same. So, yeah.
one of the interesting things that I noticed in this paper that I hadn't read anywhere else was he uses the term the same biochemical phenotype, which is pretty much what you were referring to. They have a very different genotype and they have a very different history, but they have a similar biochemical phenotype. So they all look pretty much alike, but they have, which took why it took so long until the late 1970s before anyone figured out that they had been separated for 3 billion years or so.
Yeah, I mean the basic is the same, right? They use ribosomes. They have DNA. The mRNA is different in prokaryotes and eukaryotes. Also the cell wall. Bacteria have a cell wall. I think archaea it's also different, the cell wall between bacteria and archaea. Eukarya don't have a cell wall in this way, except for plants. They have a cell wall again, and fungi have also one that's different from plants. So yeah, so there are biochemical differences, but the basic is the same.
Is this related to what he says here in the paper about halophiles whose cell wall contains no peptoglycan?
Yeah, yeah. So they are in the bacteria. They are gram-positive and gram-negative bacteria. So the structure of the membranes are different. I actually right now don't know how the cell wall looks between archaea and bacteria in general. Yeah, I don't know. I don't know that right now. I think they use something else, like that. For example, fungi use chitin for the cell wall, and what was it? I think plants use lignin. Oi, 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 my botany professor would kill me right now. Yeah, cell wall of plants. Let me yeah, that is very interesting. Or cellulose, of course. Yeah, cellulose, and that is chitin for fungi. So yeah, and so there are differences. I mean, there is a function, and there's a cell wall, and but the biochemistry is the same. They use all ATP, adenosine triphosphate to produce energy, right? So
I'm going to advance my slide here. You probably won't see it, but I can reset it again. But I want it's supposed to transition to show first it tips on the side, and then we show the tree of life that are showing where thermophiles and hyperthermophiles. Can you see this now, or did it not change for you?
no, do you mean the color?
It actually says thermophiles and hypothermophiles. Can you see that on the screen?
No.
Okay, I'm going to stop share and then I'm going to reshare again so you can see that slide. Okay, now I put
yeah. I see it now. Yeah. Oh, now it's in big. Yeah. That's better. Maybe now it works when you advance. But yeah,
well,
let's.
I had to stop and then stop the share and then advance it one and then redo it. But in any case, this is a great diagram because it's so suggestive. Now we can't prove that the origin of life was with thermophiles or with extremophiles, more generally speaking. But you see where the root of the tree of life seems to be, it's all extremophiles. So, like I said, we can't prove causation, but there's a clear correlation between what appear to be the origins of life and the common root, the last universal common ancestor, Luca, and extremophiles, because all of these at the root of bacteria and archaea are thermophiles and hypothermophiles. Woese and Fox also mentioned, as I mentioned, halophiles in the paper. And interestingly to me, when they wrote this paper, basically the first thing and the primary thing they recognized were all their archaea were methanogens. So that's where they started populating the archaea was methanogens, but now we know about much more than methanogens among the archaea.
Yeah. It would be interesting. That could be a project for us later on. It would be interesting to look into that picture and then into the species when in the Earth age or epoch they emerged and what the Earth looked like at that point. So because when you see in the blue cloud here, so it seems like that was all thermophile and hypothermophile, but then there were a few branches, they branched out, which are not. So
yeah,
and that would be interesting at what time they emerged and how Earth looked like. The same for the green. So it branched out, and then also when does it this one here? What is it? Giardia? I never heard about that. Interesting. So when this occurred, and
Giardia? Are you saying
Giardia? Okay, yeah,
Giardia.
Yeah. So that would be an interesting to put a timeline on it when that was. So
this is like a static diagram that makes it look like everything at the same time. So yeah, even better if you could animate this in time and show the tree growing and just adding branches as time goes on. I would speculate that the reason we have so much diversity, phenotypic diversity among the eukaryotes is because that's where the most complexity arose, and it made it possible for that branch to become more complex and add more functions, whereas the others stayed basically dead simple.
Yeah. So in these diagrams, I think the length of each branch is proportional to the amount of differences from the last. So when you look at that, there is for the red eukarya, that's the longest branch before something new comes out. So probably that came way later, when the conditions of Earth were much better, and not much better, but more like it is today. Interesting. That would be really, yeah, that would be a nice project to see in time how that looks. Yeah, very interesting. Sorry, I interrupted you.
Yes. No. I'm going to try to advance this again. Can now advance again? Can you see the?
Yes. No, I see that now. Yes.
Oh, good. Okay, yeah. So you can give your spiel about the 16S ribosomal RNA that you were explaining to me earlier.
Yes. So we have two different ribosomes, the bacterial ribosome and the eukaryotic ribosome, the 70S and the 80S. And for each ribosome, we have two different subunits, a large one and a small one. And here we see the small one, which is the 16S, one is just the RNA, but here actually there are a few proteins too. So a ribosome is mainly, that's why it's called ribosome, ribosome, because it's an enzyme. Enzymes are normally made of proteins, but this is a enzyme what is made of RNA. So and a ribosome is basically the one thing what makes other proteins. So that is a machinery how the organism can grow and make things from the DNA. So, and as I said, there is a large subunit and a small subunit, and these subunits are consist of RNA, ribosomal RNA, and proteins. And then we have here the 16S ribosomal RNA, which is just the RNA. So the S here that comes from the scientist Svedberg, and that is just related to the speed of sedimentation. So that is a sedimentation constant. So when you have a large number like 80S, for example, so that means it takes longer time to sediment in the centrifuge, and 16S goes much faster, so it's a smaller particle. So that is just a that is what Svedberg figured out some time ago. I don't know when he lived, but I think he got the Nobel Prize for these kind of things, and I think he was the inventor of the ultracentrifuge as well. So what does it mean? They looked at these 16S ribosomal RNA, which is around, I think, 1,500, and then there's an 18S for eukaryotes, and depending on the differences here, they could recalculate or calculate back how distinct they are. So what is happening, a ribosome is critical for the organism, right? So when there is a mutation and the mutation is critical for the function, the organism dies out and that doesn't move forward in evolution. But when there is a mutation what doesn't really makes a difference in the function, then it stays, and over time you accumulate, or even you have a gain of function, you accumulate mutations which don't have a difference or have a gain of function, then you become more and more difference, which is evolution. And again, the more mutations or differences you have with a functional ribosome, the more distinct in evolution they are apart. So yeah, I hope you could understand a little bit.
Yes, that was great, and that okay. Is why Woese in his laboratory chose this particular molecule for their molecular phylogeny. It was something that was very ancient, represented in all these lineages, and it could be traced forward or traced backwards, as the case may be, to show how long they had separated, and to show that this division really did go back to the very earliest origins of life. To produce 3 branches on the tree instead of 2 branches on the tree of at the root of the tree of life, I guess you could say, if you still want to use that metaphor. Although, like I was quoting earlier from the Doolittle paper about the uprooting the tree of life and people searching for different explanatory metaphors to try to understand life. And on that same topic, I recommend this book by David Quammen, The Tangled Tree, A Radical New History of Life, in which he discusses Woese's work in detail, and those of his collaborators and the people who agreed with him and the people who disagreed with him. So it's a great account of the origins of molecular phylogeny. So I was saying earlier, can you see this now on the screen is the methanogen?
Yes.
As I was saying, when Woese was first doing his work, all the archaea that they knew about it originally were all methanogens. As you can see, it's a very simple cell with no nucleus. And here is a little actual photograph of a methanogenic archaea.
Mm-hmm.
And here we see some of the things that we know to exist in these different classes: methanogens, hyperthermophiles, extreme halophiles among the archaea, and among the bacteria, cyanobacteria, proteobacteria, green non-sulfur, green sulfur, bacteria, I don't know how you pronounce that, bacterioids,
Yeah.
thermotoga. So
yeah. Yeah.
So that was as many.
Ah.
Just a rough preparation of some slides. We'd have some images to go along with what we were talking about. So,
yeah.
And I will stop screen sharing and stop. Okay.
So yes, again, this paper is also not very long.
Yeah, it's just 3 pages, and it's a classic introducing the 3 domains of life, which is now any biology textbook you pick up today will show the 3 domains of life, and you'll learn the archaea, the bacteria, and the eukarya, and that is because of the work of Woese's lab and his collaborators. Although, as we noted earlier, he left open the possibility that there still might be other domains to find. And as you pointed out, conditions on Earth have changed. There may have been other domains in the past that have, there could have been an entire domain that has disappeared, or more than one.
Yeah.
And if there were any progenotes, or depending on whether you consider the progenotes living or non-living, because that's another question. There are no progenotes, so if they existed, they're all gone. So that's another area of research that attempting to reconstruct what would have come before and would have preceded the root of these 3 divisions of life.
Yeah, and at the end, actually, I think he it's still the question if the first common ancestor of all 3 lines was actually a prokaryote, or which was it? And yeah, maybe that was here. Yeah, the question that remains to be answered is whether the common ancestor of all 3 major lines of descent was itself a prokaryote. If not, each Ur-kingdom, which is actually also a term he introduces here, what Ur is in German, it's actually a German thing, like Urzeit is, Ur means very old, ancient, so ancient. So it means the ancient kingdom represents an independent evolution of the prokaryotic level of organization. Obviously, much more needs to be known about the general properties of all the Ur kingdoms before this matter can be definitely settled. Yes, it was 50 years ago, and I'm not sure if we know that now, or if somebody answered that question already.
Well, it still remains an area under research. Like I said, nobody's found a progenote. There have been theories of kingdoms, but there's no consensus, as it were, and there's no consensus on the order in which these kingdoms emerged, if they did emerge at the same time or at different times, or what emerged from what, etc. So there's still a lot of work to be done there for those who have the patience for molecular phylogeny.
Yeah, and maybe there is a different molecule what could be used. Maybe the ribosome is not the right one, or maybe there are different ones.
Before we started this, we were talking about how computing power had made what took years in the 70s would be done very quickly today. Now with more advanced computing power, and all computers that are available, it might be possible to use a more complex molecule that would have been not a good choice back in the 70s because of the limited hardware and the limited processing power.
Yeah. Yeah, I think we summarized the paper.
Yeah, and that is second in a series of classic papers in the biosciences, and we thank you for joining us.
Yeah. Thank you very much. Have a good one.